Okay, the PowerPoints for chapter 7 and 11 is together. Am I making some sense? Chapter 7, and you saw it on Diffusion. You saw it on Diffusion. Chapter 7, chapter 11 are combined together. The plasma membrane, same as I refer to it as cell membrane. Your textbook says plasma membrane. Thank God. So I refer to it as plasma membrane. Your textbook refers to it as a cell membrane. Same thing. Plasma membrane, cell membrane. This diagram, plasma membrane, cell membrane, is yellow, right? Yellow. Outside is cell wall. Outside is cell wall. Inside, right here, the yellow is cell membrane. Your textbook referred to it as plasma membrane. It's the boundary of the cell, of course. Every cell has a boundary. Even though some cells, like your cardiac cells, have junctions between them, right? But they have a boundary as well. They have a cell membrane as well. They're connected together. Some cells can be connected together, but again, they do have a boundary. Separates living cells from their surroundings. The plasma membrane exhibits selective permeability, allowing some substances to cross it more easily than the others. That's the experiment you're going to do today. Today, you're going to deal with a dialysis bag that will allow some molecules in, will not allow other molecules in, and then you're supposed to write a lab report. You're supposed to write down all of your classmates' results in there. So we have everybody's name on the board, and then you write down with the weight of the bag. I'll not go over that. Hang on. So that's what this is all about. What is the structure of cellular membrane or a fluid mosaic or lipid and proteins, phospholipids? Already you know. Amphipathic molecule containing hydrophobic and hydrophilic regions. Amphi, it means both. Is that right? Amphibians, animals that are amphibians, it means they live in both land and in water. Amphibious assault, United States Army, Marines. Any of you go to, are you a member of Marine, Army, anyone? I had a student in the other class. I said, you're in Army. He said, no, I'm in Marine. So I'm sorry, to me it's all to me it's all under one umbrella. To me, it's all one thing, military. They said, no, I'm not Army, I'm Marine. I said, okay, I'm sorry. But anyhow, amphibious assault, they call it. They go from sea, the soldiers, the Marines go from sea to land to attack a country, for example. So amphibious, it means you have both. A molecule, that's what cell membrane is. The head is hydrophilic, and the tail, you remember that from last lecture? And the tail is hydrophobic. Okay, so those molecules are called amphipathic molecules. A phospholipid barrier can exist as a stable boundary between two aqueous compartments and a fluid mosaic model, which I will talk about in here in a minute. You have this right here, the phospholipid heads and glycerol, and the fatty acids tail. Right here, we already talked about it. We already know, and here they are, the protein molecules embedded. We already talked about that. Here are cytoskeleton. Here is extracellular matrix. Right? You all know this. You already talked about it. You already know everything about it. Same thing here. Carbohydrates attached to it. I wrote down the name of the molecule right here, glycoproteins. Right? Peripheral proteins. They call this peripheral proteins because it's not embedded within the phospholipid. This is called integral proteins because it is embedded within the phospholipid bilayer. Am I making some sense? The difference between integral protein molecule and peripheral protein molecules. Peripheral protein molecules are usually attached right here. See, they're both of them. Either attached to phospholipid or attached to other protein molecules. And what is this, guys? What is that molecule right there? It's what? No. This one, this one, this one. No. These are saturated and unsaturated. But what is this? What is that? What is that? I think I talked about it in the past. Your cholesterol. So everything you learned in the past, now I'm adding one more piece of information to it. Cell membrane also has cholesterol in there as well. Now, plus all other goodies that I talked about. All other goodies that I talked about, cell membrane also has cholesterol in there, embedded within the phospholipid bilayer. Phospholipid bilayer embedded are cholesterols. And of course you have protein molecules. Now, glycolipid, I talked about that. This is oligosaccharide attached to lipid molecule. It's called glycolipid. Oligosaccharide attached to protein molecule called glycoprotein. Not all of the proteins in the cell membrane have oligosaccharides attached to them. Okay? Anyhow, what is the fluidity of membrane? Phospholipid is a plasma membrane, can move within the bilayer. Most of the lipid and some proteins drift laterally. Rarely, a lipid may flip-flop transversely across a membrane. What is it saying? What is it saying? That statement, I should have put these write-ups right here. What am I saying? That this membrane is fluid. It's not static. Look at this table. This table is static. It does not move. Am I making some sense? This whiteboard is fluid. It moves. Right? It does move. We are all fluid in here. We come and go. But this table is static. This, the window is static. Does not move. Am I making some sense? The door is static. Does not move. It opens and closes, but does not move. So what am I saying about cell membrane? Cell membrane, these phospholipids are constantly moving. If 1 minute this phospholipid is here, next minute or hour, I don't know the time, do not worry about the time, this phospholipid, if I put a radioactive material on this one, next minute is probably here. This protein molecule, next minute is probably here. So they constantly move. That's what, when they say cell membrane is a fluid, it means the molecules in there are fluid. Okay? Another thing, the question is, Amir, so the protein molecules move, yes. The protein molecules can come inside, move laterally, and so on and so forth, yes. Can these phospholipid flip-flop, like our politicians? Yes. Our politicians, they flip-flop sometimes? Sometimes. I don't want to blame anybody. Can this one go up here and that one come down here?
Yeah.
Yeah, they do. But how often? Not very often. That's a seldom act. That happens seldom. It doesn't happen often. So they flip-flop. That's what it's called, flip-flop. Flip-flop. They flip-flop transversely across the membrane. Rarely happens. And they call this model, you probably have seen it, Fluidity, Mosaic model. If you see mosaic, it means, I don't know, have you guys gone to fancy houses? They have a little pattern of mosaic on the floor. You know what I'm talking about. They have mosaic. And that's what this is. If somebody look from up, they see a mosaic. And of course, phospholipid embedded in between. So first people looked at the cell membrane, they saw a mosaic, they call it mosaic model. Okay, I hope I'm making some sense. That's what the term, if you come across in your readings and PowerPoints, you're reading the textbook. I hope you're reading the textbook to get a deeper, better knowledge for the material. I'm hoping then you will come across the word mosaic. It means this is called mosaic. Somebody looked from up, it looked like a mosaic. But anyhow, most of lipids and some proteins drift laterally. I talked about that. I said that. Okay, and then the flip-flop happens very rarely. How do they know the protein molecules and all that move? They took two cells, mouse cell and human cells, and they put membrane protein molecules, and they put them together, they fuse together, a hybrid cell, And of course, you had a cell like that. So they knew by some experiments, they knew those protein molecules move around. Based on that, they figured out that those protein molecules move around. What are properties of membrane? As temperature cools, membrane switch from a fluid state to a semi-solid state, like butter. Butter is a semi-solid kind of state, a solid state. Okay, so as you cool lipids, as you all know, you take, if you don't trust me, take your olive oil or the oil you have at home, put it in the refrigerator. You're not supposed to, well, I don't know. Some people do. Put your oil in the fridge, it becomes solid. As you cool it, oil becomes solid. Same as cell membrane. Our cell membrane is phospholipid. It's a lipid. You take this in the freezer, it will become solid. Cell membrane. Okay. Temperature, the temperature at which a membrane solidifies depends on the type of lipid as well. Membranes rich in unsaturated fatty acids are more fluid than those rich in saturated fatty acids. Membrane must be fluid to work properly. They are usually about as fluid as solid salad oil. Asymmetrical membrane, of course, you look at these membranes, look at them, they are not symmetry. You have that protein molecule here, you do not have it on that side. You have that peripheral protein molecule, you do not have it that side. So cell membrane is not symmetry. If I cut this in half, that portion and that portion are not alike. And that's what it means by they are asymmetry. Right here, they are asymmetry. Asymmetry, it means when you look at the mirror, that's a symmetry. Your hands are symmetry. Cell membrane is not symmetry. You cut it in half. You cut the cell membrane in half. It's symmetry. If I cut myself this way, right here in the middle, if I cut myself in the middle, is this half symmetry of that half? Yeah, sure. This half and that half look alike, but not cell membrane. Not cell membrane. You cannot cut them and then both halves. Steroids, cholesterol, I already mentioned that, cholesterol has different effects on membrane fluidity as different temperature. At warm temperatures such as 37 Celsius, which is your body temperature, cholesterol restrains, confirms, holds on, okay, confines, holds on movement of phospholipids. At cool temperature, it maintains fluidity by preventing tight packing. So at your body temperature, that's what the purpose, what is the purpose of cholesterol? I mean, it slows down that fluidity. The fluidity I was talking about, the protein molecules go from here to there. So cholesterol a little bit slows it down. Okay, so that's what the function of cholesterol is. And membrane is built by endoplasmic reticulum and Golgi apparatus as well. Those are, here is, if you look at it, you have more, this is in your, it's all written up. You have more unsaturated fatty acid. This is unsaturated fatty acid. This is saturated fatty acid. Saturated fatty acid. Saturated fatty acid. Unsaturated fatty acid. You all know what I'm talking about? Material from exam number 1. So if you have a membrane like this is porous. Porous membrane. It's more fluid. Stuff can come in and get out easier. If you have a membrane that they're all unsaturated, they're all saturated fatty acid, they're all saturated fatty acid, that's a viscous, that's a saturated, that's a viscous membrane. Pretty much does not allow much of anything comes in and out. Cholesterol within the animal cells, cholesterol reduces membrane fluidity at moderate temperature, but at low temperatures, hinders solidification. So that's the advantage of cholesterol. You have it right here in the cell membrane. What are membrane proteins and their functions? Peripheral proteins. We talked about that. Integral proteins. I did show you guys that. I did talk about that. And integral proteins that span the membrane are called transmembrane proteins. The hydrophobic regions of an integral protein consist of one or more stretch of nonpolar amino acids, often coiled into alpha helix. This needs to be elaborated more when you take cell biology. They talk about this a little bit more. But don't worry about that statement too much. I'm not going to go into details of it, to be honest with you. Here it is outside of the cell. Here it is, they're calling it alpha helix. Right here, it spans through the cell membrane, both phospholipid. And then you remember the protein molecules? Something from material from exam number 1. You have a what? N terminus and C terminus. Where is it? C terminus. What is it? What do they mean? N terminus and C terminus. Refresh my memory.
Carboxyl N.
Very good. Carboxyl N and no, that's a carboxyl N is the acid N. And then the N, acid does not have N. N is what N? Nitrogen. No. What is it? Anybody? Amino N. NH3. You remember that? Amino N. So all protein molecules have an acid N, carboxyl N, and amino N. Okay? So anyhow, inside of the cell, outside of the cell are labeled. 6 major functions of membrane: transports, enzyme activity, signal transduction, cell-to-cell recognition, intracellular joining, attachment to cytoskeletons and extracellular matrix, which I think chapter 11 will go over this. We'll talk about this a little bit. Here they are, all of them. I'm not going to talk about it right now, but as time goes on, you become familiar with it. Here, you know, like this is HIV virus, for example. Your surface of your white blood cells have recognized, this is white blood cell, this is HIV, and then what happens, the protein molecules on the surface of your white blood cells recognize HIV, and they will accept them, they bring them into the cells. But the cells of your liver do not have these type of protein, so we will not accept HIV. Am I making some sense, everybody? So the cells of your brain do not have these protein molecules, would not accept them. It's just your white blood cells, and not all of your white blood cells, certain white blood cells, CD4, CD8, they will accept them, and then it goes inside of the cell, and you know the rest of the story. They will destroy the white blood cells. Membrane is built by the ER and Golgi apparatus. So what happens, Golgi apparatus and rough endoplasmic reticulum make cell membrane right here, and the cell membrane comes and attach. Well, these vesicles, I'm sorry, let's go back. The endoplasmic reticulum and Golgi apparatus, they make up phospholipid, and inside of phospholipids you have these protein molecules, and those protein molecules come and excrete outside, but that protein molecules attach to cell membrane. And that's how cell membrane regenerate themselves over a period of time. So the cell membrane you have today is not the cell membrane you had last month. So the cell membrane, they always regenerate, just like your skin. Your skin, this is not the same skin you had 2 weeks ago. Your skin are regenerated. You shed them, that's why you take shower, you get rid of them, and new one grows back, constantly. Same of the cells from your mouth, your anus. You get rid of them every 2 to 3 days. Okay, same as cell membrane. Cell membrane, a new one comes into a place because they are made inside of the cell in Golgi apparatus and endoplasmic reticulum, and when they dump their protein molecule outside, that cell membrane attaches, that vesicle membrane attaches to the cell membrane. I hope I'm making some sense. What is permeability of lipid bilayer? Hydrophobic nonpolar molecules such as hydrocarbons can dissolve in the lipid bilayer and pass through the membrane rapidly. Hydrophilic molecules, including ions and polar molecules, do not cross the membrane easily. So what that statement is saying, what molecules can pass these membranes, it depends. If it is ions, it can pass through the phospholipid bilayer easily. It depends what type of ions it is. And sometimes, not sometimes, all the time we have protein molecules embedded here that they act like a channel, and those channels open up and close, allow water molecule, for example, to get inside of the cell and get outside of the cell. Sammy, do you have a hot date? What are the transport proteins? Transport proteins allow passage of hydrophilic substances across the membrane. Channel proteins like aqua protein. So you do have those protein molecules that I mentioned to you guys. Those protein molecules right here in this model, you don't see it, but you imagine my finger is a protein molecule. All of my fingers are protein molecules. And those protein molecules are like a channel. One of the channel that I mentioned for you guys is called aqua protein. And guess what is aqua protein allowing inside of the cell? Water. So you do have cell membrane have these channels that allow proteins and ions and other molecules in. Other transport proteins called carrier proteins and bind to molecules and they change shape, shuttle them across the membrane. A transport protein is specific for the substance sequence. Diffusion is what is a passive transport? Is diffusion of substances across a membrane with no energy investment. Diffusion, although each molecule moves randomly, diffusion of a population of molecules may be directional. What is the definition of diffusion, you guys? So here they are showing you the, today in the lab I have, is osmosis coming up? No. Okay, today in the lab, if I have a bottle of perfume here, what would happen to the molecules of the perfume? They diffuse throughout the room. Is that right? That's what diffusion is. Movement of molecules, diffusion means movement of molecules from high concentration, bottle of perfume, throughout the room, where there is not much molecules of perfume back there on the corner. So molecules are moving from high concentration to low concentration is called diffusion. But when molecules move from high concentration water molecules